Thank you all for being here. I hope you're excited to hear about sort of gene-based approaches to infer bacterial pangenomes. Before we start, I just thought I'd go over what exactly I'm going to talk about today. So I thought I would begin just by sort of going over refreshing, if you're maybe not particularly familiar with the concept of a pangenome in bacteria, talking about what that is, with an aim to kind of motivating why we need them, and that they're really essential to studying bacteria in all kinds of ways, and also why in bacteria it's maybe easier than it is in sort of eukaryotes and multicellular organisms to infer them, and how these genic methods work really well. I'll then go into detail, in quite a bit of detail, about this sort of very classic method for inferring bacterial pangenomes using gene-based methods called RORI. It's probably one of the most cited bacterial genomic methods out there. I haven't actually checked that, but it's enormously popular and it's still used today. And I'll go over sort of how it works, what it does, and actually also maybe why it's not the best method to use. And then I'll talk about some work that I've done to develop methods to compensate for where RORI sort of falls apart, and also some ongoing work, particularly in John's group here at EBI, to develop sort of further methods, particularly for these genic-based pangenome methods. Before we begin, I said, let's talk about what a pangenome actually is. Most of you are probably at least passingly familiar with the concept of a genome. And like this one here is a human genome. It's a karyotype of the 23 chromosomes that make up a human genome. And there's a lot, obviously, one could say about this, but for the purposes of this talk, I think the important thing in this slide is that these human genomes, and indeed most eukaryotic genomes, they're in some ways very nice. So each chromosome has an end and two ends even. And so there's an end-to-end kind of linear representation. And you can have, they're sort of aligned, and oh, that's the reference genome. And you can imagine from one telomere at one end of the chromosome, just reading all the DNA all the way along to the other telomere, and it makes a nice line. You can join up all those chromosomes together. Sorry, it's just my email reminding me, I think, that this is going on. You can join up all those chromosomes together and get a reference genome. And I would say today the dominant paradigm in most eukaryotic genomics is still to take that reference genome, and when you sequence something new, the sort of shorter blue lines there is the sort of mysterious sequence data. You DNA sequence something new, and you rely on the fact that you have a really nice reference to get a novel genome. And that works pretty well because the difference between the reference and the novel genome is usually not so much that you're missing things from the reference when you sequence something new. In bacteria, and this is just a picture of a E. coli genome, which I know looks a lot bigger, but do remember that it's about 100 to 1,000 times smaller than the human genome in the previous picture. That sort of let's get a linear reference doesn't work so well because the chromosome is not linear. There's no nicely defined telomeric ends. It's a circular chromosome. And even if we do sort of get the circular reference, and for a lot of species, certainly back in the day, people created really good references for, you can then sequence a new isolate with new sequencing data, and you can try and map to that reference. But we found very quickly in bacterial genomics that did not work particularly well. And the reason is some of the data that you sequence for the novel isolate doesn't actually map to that reference at all. So these red reads, I just kind of chose some from my little read schematic at random, but you can imagine that there is some part of what you've sequenced that doesn't map to the reference. So if you do a reference-based analysis where you use a reference to sort of construct or map variants from a novel sequencing, you're missing lots of data. And this was basically how the idea of a pangenome came about. It's initially from this paper. I don't know if you can see the bottom of the slide here. I don't know if I can zoom out. Anyway, it's a 2005 paper in Group B Strep, if you're interested in looking it up. And it's actually the paper where they first defined the concept of a pangenome. And, the details of this figure maybe aren't important for the purposes of this talk, but what they did was they sequenced 8 different Group B Strep, and they found, after painstakingly and with a huge amount of effort aligning all 8 of these genomes, that there were these large regions that didn't match up between them. right? So even though it's the same species, even though these are, even I think within group E strep, they were quite related genomes, despite that, there was these large regions of the genome that they couldn't match up. And that led them to sort of defining this concept of a bacterial pangenome, where, in a lineage like a species or a strain, a subspecies, for instance, the pangenome isn't just what you find in one isolate, one bacterium. It's sort of this superset where there's some elements, which they call the core, that are present in every single isolate, and then a huge array. And sometimes in some species that's more than what's in the core. In some species it's a lot less. There's a lot of variance in how big pangenomes are. But there's this accessory genome in addition to the core that some of it's unique to individual bacterium, some of it's shared between outbreak lineages, strains, subspecies, etc. And so as soon as they defined this, there was a lot of interest in saying, well, if we sequence a new bacteria, how can we get from our raw sequence data to this kind of set representation where we know what genes, what of our newly sequenced bacteria, what's shared between all 500 samples, what's present in only individual samples, what's shared between, my outbreak strains of interest. So it was a really sort of a key problem. You can imagine if I'm studying this outbreak, I need to know if there's anything unique genetically about that outbreak that isn't present in the rest of the species. So there's a lot of interest in developing these methods. And the first and biggest method I'm going to talk a lot about, and actually maybe predated, but certainly the first widely used method, Rory, was developed in response to this. And it took advantage of sort of three key technical innovations that happened in 2000 to 2015 in both bacterial genomics and actually protein database studies and sort of the protein sequence research. But the first of these was the fact that it became sort of obvious around 2010 that because bacterial genomes are so much smaller than eukaryotic genomes, remember 100 to 1,000 times smaller, at sort of bearable cost, even back then, sequencing is obviously a lot cheaper now, but even then, one could sequence a bacterial genome with enough read depth to get a pretty good assembly without any kind of reference of the thing that you just sequenced using a computer. The first reference genomes, a lot of them were done with a lot of painstaking work by hand. But the sort of shotgun short read sequencing, by about 2010, you could get a reasonably good draft genome. You would maybe assemble a quarter, a half of it in one contig, and then the remainder in smaller contigs, but that was kind of, broadly speaking, good enough. The second major technical innovation was the sort of development of these methods to very accurately predict where the genes were given a whole genome sequence. Because bacteria, unlike eukaryotes, don't have alternative splicing, introns, any of the sort of complexity of transcribed but untranslated regions in their genes, these computational methods are actually quite simple. They didn't rely on any sort of very complex or expensive computing. And you could run basically an entire bacterial genome in a relatively short amount of time. So with these two technical innovations, you could go from raw sequence data, get the assembled bacterial genome, and then go from that assembled bacterial genome to actually finding the gene sequences relatively easily, both in terms of like the amount of work required by a human, but also the amount of computational resource required to do that. The other big innovation that happened in the early 2000s were these really rapid protein clustering methods. So protein sequencing is a little older than DNA sequencing, and even by the early 2000s, I think there were quite a lot of protein sequences around, and so there became a lot of interest in developing methods to rapidly cluster these sequences. And this is the first of these methods. It's actually the method that Rory and Panaroo, the tool I'll talk about later in the talk, they both use. It's called CDHIT, and I think it's published in 2001 or 2002. And you can see that even then, it's clustering 500,000 protein sequences at 90% identity in about 2 hours. On a computer, that's 10 times slower than the sort of average smartphone in 2025. So these are incredibly fast. They're still fast for most applications today, even though there may be some slightly faster methods. And these are the three pieces that sort of were needed to come up with these gene-based pangenome methods. You may be thinking, oh, that makes a lot of sense. I think I can put those pieces together in my head. But if you haven't done that, let me do it now for you. These three things together basically let us say, if we have raw sequence data, we can sequence 10, we can sequence 50, we can sequence 500 bacteria. We can de novo assemble all those raw sequence data into assemblies. From those assemblies, we can very quickly predict the protein coding genes, and then we can translate those DNA sequences into protein coding genes and use these really rapid clustering methods to very rapidly group those protein coding sequences together, all of them so all the genes in all the genomes that we've sequenced. And even if it's 2,000 genes a genome, you're sequencing 100 genomes, that's still well within the capability of these really fast protein clustering algorithms. So in a sense, that kind of is it, right? Like that is your pangenome. You've got clusters from the protein clustering algorithm that you can go back and say, well, this gene is present in these genomes, and this one's unique to this genome. It's not clustered with anything else in the protein database. So, have we done pangenomic inference already? Not quite. So this is actually the schematic from the Rory paper, which is a little more complex than the simplified flowchart above. But it has the same major steps, right? So we get the sort of annotated assemblies and we throw them into some sort of clustering. And then there's a second different clustering. And then we do some post-processing on those clusters to make them more accurate. And those post-processing and iterative clustering steps are necessary because of two problems that complicate pangenome inference from this very, very simple clustering problem. The first of these is the fact that pangenome relatedness is not actually the same across all genes in an organism. So, bacteria are evolving, different parts of the genome are evolving at different rates. You can imagine that something that is surface exposed, that interacts with the environment, particularly in sort of host-specific bacteria, like bacteria that infect humans. That's under a lot of selection from the human immune system, for instance. And also there's pressure to generate diversity in a lot of these surface-exposed things. Whereas other sort of very evolutionary conserved regions will be under a lot of negative selection. So these protein clustering algorithms, they only allow us to cluster everything at a single threshold. Whereas in reality, the same clustering won't work for every single gene in the pangenome. So to get around this, Rory does two things. It starts at 100% identity. It clusters. Genes that are present in all isolates, if one of those clusters is present in all isolates, clearly a core gene, remove it, simplify the clustering for the next step, and repeat that process at steps of 0.5 identity. So you start at 100, you go to 99.5, then 99, then 98.5. Until you get down to 98 by default, although the user can reduce that. And then you have you found the core, hopefully. Because doing any kind of clustering not using these hyper-fast protein clustering methods is very expensive, it then uses these protein clustering methods to simplify the clustering, the next stage of clustering it does. And it uses BLASTP, which a lot of you are probably very familiar with. It's sort of a sequence search local alignment method. But importantly, it lets us generate distances. So you can take a representative from one of your really high threshold CD hit clusters and create, with a single representative per CD hit cluster, a database that you can do an all versus all blast. From the distances that you get out, you can then use more complex clustering methods, Markov clustering, to join similar CD hit clusters together. And that way you will find genes that are 99% identical across all isolates. And also you'll find genes that are maybe 90, 85. And in fact, there's no strict threshold because the Markov clustering, again, the details aren't that important, but the Markov clustering is a sort of intelligent clustering method that will look for clusters, not just have a fixed threshold. So great, we've dealt with divergent genes. The other big problem is paralogs. So gene duplication events are common in most of the tree of life. I think in plants, they're a very common method for evolution and how it happens. And it does happen as well in bacteria. And sometimes it happens very recently, and sometimes it happens in the distant past. But you can imagine how a single clustering threshold might and would cluster together two copies of a gene that are in the same genome. And really, it's two different genes functionally in that genome, and you maybe want to tease them apart because you're not going to get good results, which gene is in which isolate if you've got two of the same isolate in that given gene. To get around this, Rory finds all these clusters, these gene clusters, from its upstream steps where there's two copies from a single isolate. And it takes those two copies and it constructs a gene neighborhood graph up to 5 genes away. So it goes back to the original assembly and it says what's 5 isolates upstream and what's 5 isolates downstream from this gene. And it does that for both copies. It then also does that for the rest of the genes in the cluster and computes distance metrics. And then it uses the distance metrics to assign clusters out. So it basically takes gene clusters from its upstream clustering steps, and it splits them up based on the number of paralogs that seem to be present. And it might be the case that you have a really ancient paralog that's present in all your samples. So, a lot of sort of pillin genes in some genera are a good example of this. And it will essentially split out, the 2 different pillin genes, hopefully across all the isolates. So with those 2 things kind of resolved, Rory outputs a bunch of really, really useful output. And I think it's just worth mentioning a lot of the reason that it's used is because it's very fast to run. And it outputs all this really useful stuff that enables these downstream analyses like gene-wide association studies at the genic level, where you can say, is the presence or absence of certain genes associated with my phenotype of interest? And also this alignment of core genes, which lets you do more accurate phylogenetic inference of things that are maybe not as related. So, a lot of phylogenetic inference in bacteria is still done by read mapping against a reference genome. But if your samples are too distantly related, the problem that I talked about in the sort of first few slides of some of your reads just not mapping, there'll be a huge reference bias, and things closer to the reference will be more accurate and things further away from the reference will be less accurate because you'll be missing tons of the data. That's accessory genome. So Rory's ability to generate this core genome alignment meant that people could generate trees for much more diverse collections of bacteria. So that's all great. And when I started, oh gosh, that slide is really cut off. If I just try and do that, is that maybe more useful? When I started working on my PhD, there was a lot of excitement around using Rory and other pangenome methods. And people, though, began to notice that things were maybe looking a little unusual. And so this is the results taken from the Panaroo paper where we demonstrate how unusual this is with a TB data set from an outbreak in London. TB is widely known not to have any kind of accessory genome, right? It evolves extraordinarily slowly, and it doesn't do any kind of horizontal gene transfer, so it's a very, very clonal organism. So we expect to have a core that is essentially the entire genome and an accessory genome that should be 0. And when we ran this TB outbreak data, about 500 isolates through Rory, that isn't what we found. And we found instead that there was roughly 3,000 isolates, less than the 4,000 we expected in the core. But in Rory, there was almost 4,000 isolates in the accessory genome, despite our expectation of there being none. And so that led us to work on developing Panaroo, which uses the same kind of principles that I've just discussed about how Rory works with using the sort of assembly and annotation and protein clustering methods to generate the initial clusters, much like Rory. But it then uses population-level gene graph information to try and actively correct errors to improve accuracy. So this is an example of one of those gene graphs that Panaroo builds. This is actually of a plasmid because a whole genome would be I'll show you on a later slide, quite difficult to interpret. But essentially, the idea is for each gene in all the isolates, after you cluster it, you build a graph based on what clusters it's connected to by DNA sequences in every assembly that went into the pangenome analysis. And from that, you get this sort of representative graph where the nodes are genes and the edges are sort of intergenic nucleotide sequences that connect the genes of unknown length. The development was led by Jerry Tonkin Hill, who's doing his PhD with me at the Sanger at the time, and he was very keen to name it after an Australian marsupial, so that's why the Panaroo is the mascot, if you will, for Panaroo. What does this gene graph actually try and fix? What's going on? So we went back and we had actually a look at sort of the data and what's kind of going wrong. And the biggest source of error we found was actually from these initial processes that we rely on. In isolation, de novo assembly has the potential to be very accurate, and annotation of those assemblies also has the potential to be very accurate, often, well over 95% accuracy. But we found that even if you assume the lowest error rate of 1% per assembly and annotation, when you analyze hundreds to thousands of genomes, which people began doing sort of in the last 10 years, even those very, very infrequent errors add up to mean that your resulting pangenome analysis may have thousands of errors. Building a gene graph was how we identified these errors. And fortunately, we found that these errors, although they don't look like genes, they're not particularly random. So they happen predominantly at the ends of contigs, where there's more uncertainty for the assembler, which assembles maybe a more inaccurate sequence. And then the annotator also can then, in that sort of inaccurately assembled sequence, often find open reading frames, which aren't real. And so using that information, we built these pangenome graphs, and this is just to show you an example of what it is. This is 1,000 Staph aureus isolates, and as you can see, it's very difficult for a human to interpret, but actually a computer can process these. Modern computers can process these in fairly reasonable time and interpret them. So we're able to do these kinds of graph operations where after we've taken the original gene annotations to build this graph, we can sort of iterate over the graph and correct errors as we find them. And there's sort of two main processes that Panaroo does to try and improve the process, improve the accuracy of the pangenome output. The first is to sort of correct things that are just blatantly wrong, errors that have come in from the assembly and annotation step. And the second is to use the global knowledge of the pangenome graph to sort of improve our ability to cluster things that are extremely difficult to cluster, like highly divergent gene clusters, or to correct for annotations that have been missed. And this is done primarily by looking for things that are suspicious in the gene graph. So for the error correction, one of the easiest things to look for is contaminant sequences, because these will often be isolated on the gene graph. They don't connect to anything. They'll be found in a single isolate, and they're like their own subgraph, distant from the rest of the isolates, no connections in nucleotide sequence. So we just delete those because they're very obviously contamination. The other thing we look for is genes at the ends of contigs that are not connected to anything else in the gene graph and don't have population level support, and we simply delete those as well. When we find situations like this in the graph, we look for bubbles where a string of linear, like 2-degree nodes are connected by several nodes. We can then go and look at the DNA of these sequences and say, is this some sort of translation error because of a frame shift mutation? So is there something going wrong in the sort of clustering or the pre-clustering step that meant that these things are not getting clustered when they should be? And by using that DNA comparison, because we can do it much more cheaply, we only have to look at these two nodes instead of all the nodes across the graph, which would be computational unfeasible. We can then collapse these two nodes as well. So that corrects a lot of the errors with the pangenome graph. But then we can actually improve gene clustering by looking for larger bubbles like this. And again, because we're not doing all versus all clustering, we're only looking at local areas of the graph, we can go to much lower thresholds and we can look at DNA and say, do these gene families or do these sets of gene clusters that Rory might report as individual genes, are they actually homologous at a lower threshold? And if yes, we can then collapse those into a single genuine cluster. The last sort of accuracy improvement Panaroo does is it looks for instances where annotation fails in the other direction. I said, annotation is very accurate, and the predominant source of error is these sort of incorrect annotations at regions that are poorly assembled, and that's true. But occasionally, either due to sort of genuine biological variation or sometimes like technical error, annotators will miss a genuine DNA sequence. And again, because we're using this sort of global graph, we only need to look for instances where there's an isolate that has these 3 upstream genes and these 3 downstream genes, but for some reason is missing this gene in the middle. And because we can go back to the original assembly file and pull out this intergenic DNA, we can then look for that DNA in this cluster. We can look for this cluster in that DNA and say, can we find it and correct something that the annotator has missed? So all of that should hopefully improve the pangenome inference. And to give you an idea of sort of what that does to the graph, there was the original 1,000 Staph aureus isolates before any of the Panaroo graph methods were applied to it. And here's the graph afterwards. So you can see that a lot of these sort of large, messy regions in the graph are corrected and are basically very clean loops of sort of 2 degree or degree 2 nodes. So genes that are connected to a single upstream and downstream gene. Of course, there are regions in the genome that are full of sort of mobile genetic elements, and these will cause these sort of hairballs in the graph, but there's nothing really that can be done while still maintaining accuracy. It is possible to go and manually edit the graph if one's interested, but computationally, it's impossible to sort of do that accurately. Just sort of for some quantitative assessment, does Panaroo improve the accuracy relative to Rory? We just introduced errors in otherwise perfect assemblies and compared the pangenome runs of all these pangenome tools listed here, in perfect assemblies where there were none of these errors, and then assemblies where we'd introduced these errors. And you can see that Panaroo deals with contamination in a much lower overall error rate and fragmentation. It's still not perfect. So if we go back to that example of the TB outbreak in London, the Panaroo still reports a small accessory genome. Most of these isolates most of these accessory genes are due to the fact that not every isolate is sequenced perfectly. So there will be genes that are simply missing from the sequence data. And also, of course, there's often also an error or two, like some contamination that's consistent across many isolates. We don't have the confidence to delete it because it may be genuine, those kinds of things. But overall, we're getting essentially much more of the core, pretty much all the 4,000 genes we expect, and far fewer accessory genes than error or most methods that don't try and actively correct for errors. The global pangenome graph also allows for some additional analyses. So like I mentioned, there's been a lot of interest in doing gene presence absence association studies, typically for things like antimicrobial resistance data. And the Panaroo gene graph actually lets us do that in a little more detail, where we don't necessarily rely on simply gene presence absence. We can look at sort of structural variation in the gene graph. And that plasmid I showed you earlier is an example from the Panaroo paper where we found this example of a plasmid in Ghana where the structural variation is a sort of a different version of the plasmid, and it picks out these sort of antimicrobial resistance elements. This is a resistance plasmid in red, but in blue it didn't pick it up. And if you just did the association on gene presence absence, you wouldn't necessarily find this because you don't have the statistical power that you do when you're looking at the structural variation. So that's was published in 2020, and I think it's seen a reasonable amount of use. But there's still ongoing work to sort of extend these methods even further. Since it was published, I actually did some work to add this additional code to Panaroo, where you could not only sort of do the Panaroo analysis, put in your draft assemblies with annotations, you could actually take the final graph Panaroo outputs and output sort of accurate annotations downstream. So after you do the pangenome analysis, sort of corrected, more accurate annotations. If you're interested in doing that, though, my colleague Sam is in 3 weeks going to give another seminar in this series where he talks about sort of combined methods to do both those things at once with the nucleotide level graph and how nucleotide level graphs can maybe improve that process even further. So I encourage you to register and check that out. There are also other methods that are working on, that people in the group are working on. This is something I've been working on for the last couple of years, where some of you may know that lots of recombinant bacteria, phylogeny inference relies on us having an accurate recombination-free alignment. And so there's a lot of these methods to detect recombination, and most of them will only work on very closely related bacteria where you can do reference mapping. Currently, there is a public development version of Panaroo where we can use pairwise comparisons within the pangenome to remove recombinant sequences, and that will improve tree accuracy enormously. We're in the process of testing that, and the initial data is very promising. But it also means that we can do it for much more distantly related isolates. So it's not just anything where we can get a reasonably good reference mapping, but if you can do a pangenome analysis, you can remove recombination from that and get an accurate recombination-free tree. Something a lot of you may have been thinking throughout this presentation is, well, what about intergenic regions? And that has historically been sort of a big weakness of these genic methods, where we remove essentially the intergenic regions to do the protein-based clustering to infer an accurate pangenome. And Harry Thorpe, who I worked with in my previous fellowship during his PhD in 2018, he published this method downstream of Rory to extract intergenic regions and do a Rory-like analysis on the intergenic regions. That was in some way limited because in bacteria, intergenic regions can be so short that they're almost impossible to cluster. So I've been working on a method which is still currently in development, but basically using, again, the whole pangenome gene graph that we get from Panaroo and that we build in Panaroo, it's easy to, instead of trying to do a global clustering problem, pull out specific intergenic regions and go from this kind of gene graph to just inserting, as you can see, if you can make out the text labels, we're just inserting nodes for those intergenic regions between the genes. And that greatly simplifies the analysis because you don't have to do this sort of global intergenic region clustering. Finally, especially here at EBI, we're always interested in taking things up to the next order of magnitude in terms of scale. So, currently Panaroo will kind of work up to ideally 10 to the 3, maybe 10 to the 4 genomes, and that sort of order of magnitude. So I think the largest we've done is coming up on 20,000 isolates. Can we get that to hundreds of thousands or even millions of isolates? The problem is that the initial graph Panaroo builds is too complicated. So all those graph operations I described previously, where we talk about, can we go through and correct errors? Can we collapse divergent gene families? Can we find missing annotations, like genes that the annotator missed? All that kind of thing becomes much too computationally expensive if you build this initial graph with 10,000 isolates. So there's been some interest. Jerry wrote a simple method to merge clean graphs, but a PhD student here in John's group is actually working on new and improved methods to take gene graphs, different Panaroo gene graphs, and merge them together to get pangenomes from much larger isolates. So that is pretty much all of my talk today. Hopefully I've convinced you that pangenome inference methods are essential basically to analyze bacteria, and that also that we can do it in maybe a sneaky or computationally much cheaper way using gene annotation, de novo assembly, and these highly rapid protein clustering methods. These aren't entirely straightforward. We need to do some post-processing on the clusters, and in particular, that we need to correct errors that arise from these sort of very, very low frequency errors in the initial input, but when grouped together become extremely prevalent. Thanks so much.
All right, thank you very much. Thanks, Neil, for the wonderful presentation today. And thanks everyone for joining. And I can say our audience was very, very engaged. So I have loads of questions actually. So
Awesome.
let's see how many we can go through.
Yeah.
Yeah, we have enough time to go through, I think, most of them. The first question, I think you addressed it towards the end of your talk already, but if you have any more comments about it, you can say. It's about how about non-coding sequence in bacteria's genomes? So I think you already talked about intergenic regions.
We're working on these methods to add intergenic regions, and we're hoping that they'll be ready relatively soon, sort of by the end of the year for public release. There are obviously also methods that do pangenome analysis at the nucleotide level in bacteria, but they often struggle to scale. So depending on sort of your interest, it may be more straightforward to do a gene-based pangenome analysis first and try and add intergenic regions post hoc.
Great, thank you. Some people say Panaroo is better than Rory. Do we know why? Or is that true?
I mean, I as a Panaroo developer, I'm obviously not an unbiased source for that kind of question. I do think the active error correction in Panaroo means that it's much more accurate, or the accessory genes in particular are much more accurate. Depending on your interest, like some people do pangenome analysis because they want a tree. If your interest is, I want to run a pangenome analysis so I can get a core genome alignment, so I can get a tree, Rory is probably accurate enough for your purposes. If you want to run a pangenome analysis because you're interested in finding sort of mid-frequency accessory genes that you think may be responsible for some phenotype of interest in your study sample, in that case, I think the accuracy issues that Rory faces, particularly with these sort of erroneous sequences, you'll have to sift through lots of errors, and also often it will split core genes that are too diverse because it doesn't have the sort of collapsing, the gene family collapsing function in the graph. That kind of thing, Rory is probably not going to be accurate enough. I hope I answered that without taking sides too much.
Right, so next one is in pangenome analysis, how do we distinguish diverse gene variant from an accessory gene?
So I think this actually kind of perfectly leads on from the previous question. I think in Rory that is almost impossible because it doesn't have the context of the entire graph. So in Rory, a really diverse gene, because I mean, if it's not a paralog, it doesn't even look at the neighborhood. It just relies on the clustering. So a really diverse gene would just be split into two clusters. So you would have no way of distinguishing that. In Panaroo, during the graph correction steps, we look at the graph and we say like, oh, these two genes are always in the same place, and do they look similar at lower thresholds? So we go through the graph and we find each location where there's lots of genes in the same place, and we say, do these genes actually look similar at a much lower clustering threshold, which is specifiable by the user. I think we go down to 80 or 70. So in that sense, we should be able to collapse that diverse gene family into a single gene in the output, which you wouldn't get with Rory. In Rory, it'd probably be two genes, so you wouldn't be able to distinguish it. Whereas in the Panaroo output, you should be able to find like, oh, this is a super diverse gene, and here are my accessory genes, and it should theoretically all work very nicely.
Okay, thank you. If the copies of genes are distantly related, why would they show up together in clustering? Is it because of proximity to each other?
So if copies of a gene are distantly related, they often won't show up in the same cluster. I think it was, was it around the paralogs discussion? Yeah, I think so. Sometimes paralogs, like, so I study upper respiratory tract bacteria, so those are all my examples. But in Neisseria meningitis, for instance, there's the pill E gene. There's often lots of copies because it rotates different cassettes from within its own genome. So it'll have one copy of the pill E gene, several copies of pill S that are like the same sequence, but it keeps like 4 or 5 copies. So these are, in a sense, distantly related in that it's had that for a long time. Like this is a very old system, and I think it's probably found in multiple members of the genus, so it sort of is from the common ancestor of Neisseria. But because, they still serve the same function, they still do the same thing, it's just advantageous for it to keep multiple copies to preserve variation. They're related enough that in lots of clustering methods, they would fall below kind of whatever threshold, 92% identity threshold. And so even though they're paralogs, they would initially be clustered together. And so Rory would split them out, but in Panaroo, I think we find that we split them out even more accurately because we use the entire context of the graph.
How can pangenomics be used alongside GWAS to identify genes linked to disease in infectious strains affecting humans?
Yeah, I think there's sort of multiple levels at which you can do this. I think so. If you're already interested in doing a pangenome analysis, you could run a sort of gene-based pangenome inference method like Rory or Panaroo. And with Rory, you'd get the gene presence absence. And again, maybe you'd want to prefer Panaroo in this case because the accuracy of the gene presence absence will be very important for your GWAS. So if you take the gene presence absence of, say, Panaroo, you can then associate the presence or absence of genes with your phenotype of interest. And there are methods like SCORY or SCORY2, which are explicitly designed to do just this kind of GWAS. Can we associate the presence or absence of genes with our phenotype of interest? But PySeer will also support these kinds of things as input. So PySeer, you can do a GWAS on gene presence absence files as well. In a sort of more advanced sense, if you think that the variation isn't genes, or as Panaroo, you can do sort of structural variation in the gene graph. That's a thing you can do in Panaroo. But if you think it's not genes or gene graph structural variation, but it is some sort of nucleotide change, you can still use a pangenomic approach by, instead of doing reference mapping, finding unitigs. And that can be quite computationally expensive, depending on how big your sample collection is. If you have sort of 10,000 samples, it will take a very, very long time to find all the unitigs. But if you find all the unitigs, you can then do sort of GWAS on unitigs, which should give you more resolution than the simple gene presence absence. I should also say that if you run IGR methods like Piggy, you can run Piggy downstream of Panaroo as well. So if you want to run IGR methods today on Panaroo output, you can do that. You can also, of course, do a GWAS on the IGR gene graph, so the presence or absence of IGRs.
Great, thank you. How do you distinguish a poor assembly from a true contaminating sequence? For example, if your assemblies have many contigs.
Yeah, so we do that basically by population level information. So if I go back to not this slide, but the one after it. So this is how we identify contamination, right? And it's not simply like we don't just look for small contigs. We look for small contigs that are unique to a single isolate. And those are almost overwhelmingly likely to be contamination. And even if they aren't contamination, they are indistinguishable from contamination. So they don't typically add any useful information for you, the downstream person who has to interpret the data. Of course, you can run it without deleting these, but those are the things that we think are suspicious enough that we simply delete them from the output. The problem, of course, if you look at some of these graphs, the clean graph Yeah, we actually do still have some small things, some of which are probably contamination. And these are because that some contamination is not random. So in this case, it's a Staph aureus samples. Where you sample the Staph aureus from, you're very likely to like accidentally sample some of something else. And so you get the same thing. And so these are sort of well-supported contamination. Those are sort of, in a sense, indistinguishable from small rare contigs. So we don't delete those because they're more likely to be real than something that's only present in one sample and looks like this.
Great, thank you. Next question starts with a compliment. Great talk. Thank you, Neil. Could you comment on approaches for inferring pangenomes from metagenomic or microbiome data, where the analysis isn't focused on a single bacterial species, but a mixed community of genomes or MAGs?
Yeah, that is a really, really difficult problem. I don't know that I'm not personally aware of like a single best method to do this. I think lots of people are interested in developing ways to do this at the nucleotide level. And so there's a lot of development kind of going on in that field. I know it's possible to do it species by species. And in a sense, that may be the approach. And using sort of these merging methods, it may be to use genic methods anyway, to do sort of something from the Rory family of methods. That would probably be the approach I would take. So you start by doing it in one species, and then you take the two graphs and you try to merge them, and you'd have to do it at a much lower threshold because the sort of percent identity is going to be quite low for very distantly related species.
Thank you. Can you speak about Panaroo's error rate when using short read assemblies versus assemblies made with long reads?
We've not done any sort of in-depth benchmarking about that. I mean, higher quality assemblies almost always are more accurate in pangenome analysis. Yeah, I'm trying to think if we've ever really looked at that in detail. I think it will be slightly lower, but if you run Panaroo in like the strictest error correction mode, so we give three options, which you can additionally modify, but as sort of defaults, you can run it in strict, moderate, and sensitive. Sensitive doesn't delete anything, so it's only useful if you have sort of a very strong a priori reason to think that there's a very rare gene that you're interested in. But certainly in strict, I don't think there is like an order of magnitude difference in accuracy. It's the accuracy between short read and sort of long read or hybrid assemblies is comparable, I'd say.
Okay, thanks. Next one is about the method comparison. When I started my PhD, I was a bit naive and didn't know about Rory or Panaroo. Instead, I did my own annotation and clustering with Bacta and OrthoFinder before identifying my region of interest and building a gene-based pangenome graph of a small region of the genome. Could you comment on how this approach and the clustering done by OrthoFinder compares to Panaroo?
I think it's a lot more work. Like that sounds like a ton of work, but I don't think it'd be less accurate. I think if you're interested in a specific region, that will work really well. And yeah, you've maybe just you could have saved yourself some work, but it might have taken a long time to find and identify the pangenome graph as well, especially if it's a highly mobile region. So no, that sounds like it was a very good idea.
Can you recommend any resources for practical training on this material or something that you presented?
Practical, I mean, most of these tools will have example data sets. We tend to think of the Panaroo documentation as pretty good. So you could try and go through that and run the examples. I'm not aware of anything else that sort of will take you through a lot of it. It's not very complicated. It's just basically like command line if you're familiar with that, you should be okay to install and run Panaroo very easily. But like there are, I think EBI offers some nice courses for sort of basic bioinformatics, which may help. Other than that, I mean, certainly we try to be quite responsive in the Panaroo GitHub. So raise issues, we try and get to them. Sometimes it takes us a week or two, but we do try and get to all of them.
Great, thanks. Um, any suggestions about currently available tools or methods that you can recommend for handling recombination in E. coli?
Um, Depending on how diverse your collection of E. coli is, it makes a huge difference. I think like the sort of classic method, if you're looking within like NST broadly, I would say for E. coli, gubbins or clonal frame mammal gubbins will probably run a bit faster. If it's super diverse, you can run a pangenome analysis, and FastGear will work on the individual gene alignments. But then again, you're doing gene by gene analysis. It's public, but it's still in testing. Obviously, the method I talked about just now, and also some tools to sort of help you run FastGear at the gene level. But yeah, I think the more standard approach, if it's like a single ST, is to do a whole genome pseudo alignment and use gubbins or clonal frame ML.
Great, thanks. Uh, could you clarify how Panaroo handles highly similar paralogs genes to avoid merging distinct genes or splitting true variants?
Yeah, so Panaroo, basically, unlike Rory, which sort of uses the gene neighborhood, like the very local gene neighborhood, we have the whole graph. So we do kind of the same thing, but basically, we just we take a thing that's a paralog and we just pull the graph apart. If you can imagine, like the graph will be collapsed in that node because it's got edges, both paralogs will be in that gene node. So it will have the path of the one paralog and the path of the other paralog, and they'll be together. And we just go, oh, there's two copies from a single isolate. Let's pull that apart. And can we then easily match with the other isolates? Is it very straightforward? If it isn't straightforward, we do sometimes I think use DNA to identity to try and match it up. But it on the whole, I think produces much more accurate paralog splitting than Rory, because the gene neighborhood can sometimes be very convoluted in regions where there's a lot of mobile elements.
Great, thanks. I've used Rory before with a set of 70 bacterial genomes of the same genus. I noticed that playing around with the thresholds can really make a difference. I wanted the core genes to build a phylogenetic tree. Best results I got were with 95% limit for core genes and 70% identity for clusters. What threshold do you recommend also for Panaroo? I'll be happy to try it out.
Uh, this is like extremely specific to your genus of interest. There are some genera that are like super diverse, like maybe should be multiple genera diverse, and there are some genera that are like a single species. Um, it's impossible to say a priori. I think in Panaroo, you could the sort of thing you could do to sort of assess thresholds more easily, other than just looking at the output, is you could run them and then have a look at what the graph looks like. And does the graph look like two graphs, or does it look like one graph? And then you would have some idea of how good your threshold is overall. I mean, the sort of 9270 sounds sensible, broadly speaking, but without knowing the specific genus, and even then, it's very difficult to comment on the numbers specifically.
Thanks. How do gene-based pangenome inference methods handle paralogs genes or gene families that have high similarity? Do they risk merging distinct genes or splitting variant of the same gene? Probably it's a similar question you answered very.
Yeah, I think basically, you know, Rory uses the neighborhood, and in Panaroo we use the whole gene graph. And I think the latter approach is probably more accurate.
Do you recommend a way to build a gene presence or absence matrix using the Panaroo output? for ease of interpretation of a reader?
So we output like 3 different versions of the gene presence absence matrix. 2 of them are meant to be kind of human readable-ish in Excel. I think they're called gene underscore presence underscore absence.csv and gene underscore presence absence, with the underscores rory.csv. Those are sort of human readable. And then we also output an RTAB file, gene presence absence.rtab, and that's meant for computer reading, basically. So you can very easily read that into R or pandas or whatever you like doing statistics in.
Okay, I think a similar output kind of question. Does Panaroo build a phylogenetic tree of isolates that you input?
No, we don't do that by default. We do output the alignment, and you can run that either with Panaroo or you can run it separately after you do the pangenome inference bit. And we do have a lot of recommendations for how to run the tree, but you do have to type in another command yourself to do it.
Does the graph approach could distinguish when a small fraction of the population, 15 to 20%, has gene insertions in a code region, or will it be removed?
That is, yeah, it kind of depends. So I think basically, is this insertion, like, is it the insertion of an entire gene? If it's the insertion of an entire gene, that would be very easy to detect with these genic pangenome methods. If you're looking for like the insertion of a short string of nucleotides within a gene, you would have to, I think, have a good idea of which genes you were looking in order for it to be simple. Because after you run the pangenome analysis, you can then get alignments of every single gene. And if you know which gene you're looking for, yeah, it's quite easy to look in the alignment of that gene and say, oh, look, these 20 isolates have an insertion of 20 nucleotides. But if you're looking through all like 5,000 genes of the pangenome, it's a lot harder to say, like, this is, the insertion I'm interested in, because there's probably quite a few insertions.